All right. Hi, everyone. Thank you so much for joining Grand Rounds today. We have a wonderful talk today by one of our very own, Ajeet, who's here to present on the hidden inequities in data using an anti-racist lens to reassess race in clinical algorithms. I wanted to begin with a bio from our presenters today. So Dr. Ajeet Singh is a third-year internal medicine resident in a Highland hospital. He received his MPH from Harvard and MD from Loyola Street School of Medicine. He has extensive experience in both organizational medicine and clinical informatic projects, often undertaken within a health justice perspective. He will be pursuing further clinical training next year in clinical informatics fellowship. In his free time, Dr. Singh enjoys music production, Muay Thai, and drone flight. So very exciting. We also have Nate Jerkins, who is a hospital medicine physician and core faculty for the internal medicine residency at Highland Hospital. He is broadly interested in population health, healthcare communication, and the intersection of technology and care delivery. Outside of the hospital, he enjoys traveling, riding, and trying to avoid injury on the soccer field. And finally, we have Dr. Chris Coontz. He's a former core faculty member at Highland Hospital and currently works as a hospitalist at Cleveland Clinic in the Indian River in Vero Beach, Florida. So let's please welcome Dr. Ajeet Singh, Nathan Jurgens, and Chris Coontz.
Thank you, Alejandra. So today we'll be discussing how certain forms of bias and discrimination have presented themselves through the technologies we use and the practices we teach. Some of these biases have deep roots in racist perspectives, which have led to patient harms that our communities disproportionately face. In some ways, for many of us at Highland, choosing to work in a county hospital is a form of resistance against these forces. Nonetheless, we'll be uncovering ways in which, even with our best intentions, we may have perpetuated some additional harms. Some of these topics can trigger strong emotions, and so I want to encourage everyone to practice self-care during this conversation. So let's begin. Today, we'll be centering our conversation on this 2022 paper by Fawzi et al., Racial and Ethnic Discrepancy in Pulse Oximetry and Delayed Identification of Treatment Eligibility Among Patients with COVID-19. Over the hour, as we look at how pulse oximetry functioned in patients of different racial backgrounds, we'll start to unpack the factors that enabled these discrepancies to persist and review how those factors are situated in the broader context of racism in healthcare. As we close, I'll discuss an anti-racist approach we can apply to various layers of this problem. So let's begin with an understanding of how pulse oximetry works. This was designed to measure oxygen saturation by illuminating the skin and measuring changes in light absorption of oxygenated and deoxygenated blood using two wavelengths. The ratio of absorbance at these wavelengths calibrated against measurements of arterial saturation to establish the oximeter's approximation of arterial saturation. The waveform, available on many pulse oximeters, assists clinicians in distinguishing an artifact from the true signal. The concept was applied to create multiple sites of light transmission, reflectance, and detection, suitable for the finger, toe, earlobe, and forehead. But in the early literature, certain limitations were clearly identified that could impact the detected light. One example is low perfusion states, which include low cardiac output, vasoconstriction, and hypothermia. Operator proficiency is also often overlooked and represents a critical layer of communication between the device and the provider. So SpO2 values are often reported and documented without adequately assessing these limitations. Today, I want to take a closer look at skin pigmentation. Deoxyhemoglobin and melanin are the primary light absorbers at these wavelengths. And so measurements of light will vary with skin pigmentation depending on tissue perfusion, probe location, shape, and color. So ever since modern pulse oximeters were deployed widely in the critical care setting, there have been concerns regarding its accuracy in Black patient populations. A 1990 study performed on mechanically ventilated patients found that for white patients, a pulse ox target of 92% was reliable in predicting satisfactory oxygenation. However, quote, in Black patients, such an SpO2 reading was commonly associated with significant hypoxemia, and a higher SpO2 target, 95%, was required to ensure a satisfactory level of oxygenation. 15 years later, in 2005, a UCSF study comparing 3 different pulse oximeter companies found that not only did dark skin pigmentation result in an overestimation of saturation, but that this overestimation was greater, especially at lower saturations across the 3 tested devices. 15 years after that, in a 2020 NEJM study from the University of Michigan, researchers looked at occult hypoxemia in white and Black patients across 178 hospitals. They defined occult hypoxemia as when a patient has a pulse ox reading between 92 and 96 with a concurrent arterial saturation of less than 88% with an ABG. They found that Black patients had nearly 3 times the frequency of occult hypoxemia that was not detected by pulse oximetry as white patients. And then, less than a year later, researchers sought to understand, does hidden hypoxemia result in different clinical outcomes across racial and ethnic subgroups? In this study, they defined hidden hypoxemia a little more narrowly, but by looking at nearly 150,000 encounters across 215 hospitals, they found that all racial and ethnic groups experienced significant occult hypoxemia, which was greatest in Black and Hispanic patient populations, and that for Black patients, hidden hypoxemia had a significant higher in-hospital mortality than patients without hidden hypoxemia. These tools were designed by using light against the skin. And after 31 years of studies, it begs the question, whose skin was this technology designed for and tested on? Why haven't these problems been addressed? And what has been the magnitude of this oversight? And on the left, we have the 2005 UCSF study showing performance across the 3 devices. And on the right, we see the 2020 Michigan study showing how much more occult hypoxemia Black patients experienced than white patients. So as we reflect on the COVID-19 pandemic, why is this literature around the accuracy of pulse oximetry important? In October of 2020, the FDA approved remdesivir for the treatment of COVID-19, and dexamethasone's approval came shortly after. Eligibility for treatment was contingent on an approximation of their oxygen saturation, and thus pulse oximetry was key to guiding medical decision-making. What are the consequences of incorporating a technology with multiple layers of risk and failure that has been found to consistently overestimate its results across patient populations, potentially contribute to higher in-hospital mortality, and function in a broader context of operator-dependent performance, in which the lead placement and waveform interpretation increase variability of underlying device functionality? So that brings us to the 2022 JAMA Fawzi et al. Paper that I'd like to discuss today. This was a retrospective cohort study conducted at Johns Hopkins, which collected clinical data from 5 hospitals over the course of the COVID-19 pandemic. Nearly 7,500 patients were segregated using self-reporting race and ethnicity as a surrogate for skin tone. The exposures were whether they had concurrent measurements of oxygen saturation in arterial blood and pulse oximetry within 10 minutes of each other. The primary outcome was just the identification of occult hypoxemia using the same thresholds as University of Michigan paper earlier, and they added a second outcome to highlight its interaction with the COVID-19 pandemic by exploring the association of occult hypoxemia with delays in the time to recognition for eligibility of COVID-19 treatment. Notably, each racial and ethnic group had a proportional number of patients who required mechanical ventilation, and it was a fairly well-representative sample. Let's look at the primary outcome. On the x-axis, we can see each pulse oximetry result from 88 to 96, and the y-axis is the difference between the arterial saturation and the pulse oximetry result, the clusters of results showing by color the different racial ethnic groups. What we find is that for Asian and Black patients, the pulse ox consistently overestimated oxygen saturation for all SpO2 readings between 88 and 96. Meanwhile, for white patients, the pulse oximetry consistently overestimated oxygen saturation for all of those same readings. These differences do not vary by device, whether they were using nasal cannula, high flow, or mechanical ventilation. They then used an adjusted linear mixed effects model. This adjusted for age, sex, Charlson comorbidity index, certain time-varying vital signs such as MAP and temperature, a number of lab results, and the type of oxygen device. They compared all racial and ethnic groups to white patients. Just note, the figure may appear misleading where it says SpO2 underestimates on the left. That's relative to the white population's values. So if we just look at how the x-axis is labeled, we can interpret this as following. For Asian patients, the SpO2 overestimated arterial saturation by 1.7%. And for Black patients, the pulse ox overestimated arterial saturation by 1.2%. And then for non-Black Hispanic patients, it overestimated arterial saturation by 1.1%. This was all by significant degrees. Notably, the Asian group had just over 1/8 the number of patients as the Black population, and although we see a greater magnitude mean difference, it bears a broader confidence interval, but is nonetheless significant. Let's move on to thinking about the secondary outcome. What consequence does this have on recognizing patient eligibility for guideline recommended therapies? They took the adjusted linear mixed model and applied it to the original patient population's pulse oximetry values in order to predict what the approximate arterial oxygenation was, filling in for gaps where patients may have otherwise not collected ABGs. Of the total patients, nearly a third of them, or 1,900, had a predicted arterial concentration of 94 or less before either a pulse oximetry value of 94 or less was recognized or before oxygen was started. That means that almost a third of patients were not appropriately recognized as being hypoxic at some point and thus not recognized as eligible for treatment in a timely fashion. Breaking this experience down by group, compared with white patients, Black patients had a 29% lower hazard of eligibility recognition, which means that for every 100 white patients recognized as eligible for treatment, only 71 Black patients were recognized as hypoxic and eligible. And for non-Black Hispanic patients, only 77 were recognized as hypoxic and eligible for treatment, with no significant difference between Asian and white patients. Let's look more closely at this breakdown. The majority of our pie chart represents patients who were appropriately recognized as hypoxic. The cut of the pie represents the third of patients who experienced predicted hypoxemia. And then within this cut of the pie, we see nearly a quarter of patients who were predicted to have hypoxemia, but never actually demonstrated hypoxia on pulse oximetry, nor were they initiated on oxygen. These were patients who were truly eligible, but were never recognized as eligible for treatment and effectively went without treatment. We can look at this breakdown by race and ethnicity here. Over half of these patients were Black and over a quarter were non-Black Hispanic. For the remainder of the 2/3 who were predicted to have hypoxia and were eventually identified appropriately, how long was the delay? There was no significance in the difference in treatment delay between Asian or Hispanic patients and white patients. However, compared to white patients, Black patients had a significantly higher median difference in time to recognition of eligibility by 1 hour. So I want to touch on some strengths and limitations of this study. It sampled a large and more diverse population compared to some of the prior pulse oximetry studies that have been performed. And for these patients, a substantial amount of clinical information was available and analyzed, not just arterial and pulse oximetry, but all the covariates that powered the model. Also, there was a common underlying disease state. However, some limitations. Most significantly was the fact that they used self-reported race and ethnicity as a surrogate for skin tone, which is a poor proxy given that the heterogeneity of skin tones across racial and ethnic groups. Secondly, the time to eligibility recognition underestimates the actual delays in treatments that patients face. These are further influenced by variations in clinical attention, frequency of pulse ox checks, time to medication ordering, medication availability, and medication delivery. And then lastly, we have a limited understanding of the harms that resulted from unrecognized or delayed recognition and treatment eligibility. For patients who effectively went without treatment, we have evidence demonstrating the increased mortality risk in those populations, but this was not directly measured. We can speculate on ways in which delays in recognition could increase mortality risks, but this was not something that has or will likely be studied in the context of COVID-19. And so the impact of these delays may not be something that we can effectively approximate. So in summary, Fozzi et al. Identified a persistent overestimation of arterial oxygen saturation among Asian, Black, and Hispanic individuals. This overestimation of oxygenation resulted in a systematic misclassification of patient hypoxia based on racial or ethnic self-identification. This led to a systematic failure to identify Black and Hispanic patients who were likely qualified to receive potentially life-saving COVID-19 therapy, and it produced statistically significant delays in realizing the guideline recommended threshold for initiation of therapy among Black patients compared to white patients. If we accept the conclusions reached by the analysis done in this paper, this is an unacceptable finding. And it's imperative to put these race and ethnicity-based findings in the broader context of the COVID-19 pandemic and reassert for all of us that in the first 18 months of the pandemic, Black, Hispanic, American Indian, Alaskan Native, and Native Hawaiian Pacific Islander groups experienced higher rates of COVID-19 infections, hospitalizations, as well as mortality compared to white patients. So I want to pause and reflect on this. At the start of the pandemic, before the risks of COVID-19 were well detailed, and before there was any sign of a vaccine, we followed the research happening at the time and with cautious optimism took up the guideline-based treatment protocol with the hopes of preventing significant morbidity and mortality. This was a difficult and stressful time. And I want to give space to some of us who are now wondering, did I turn someone away who needed treatment? Should I have not sent that patient home whose pulse oximetry was straddling the threshold of normal? And did that patient instead need to be admitted and started on potentially life-saving treatment? I want to take a moment to acknowledge this guilt that can emerge after reading such findings. As clinicians, we embark on intensive training so that we can focus on doing the best for our patients, and we put significant trust in the systems and institutions around us to support us in protecting patients from harm. What we saw in the experience of using a flawed technology in a critical workflow was not a reflection of any individual's failures, but rather a failure in the system around us. It is possible that had we reconciled with the vulnerabilities of the technology more intimately at the time, that we may have deliberated on ways to better protect our most marginalized communities from being undertreated. Later in this presentation, we will look at some approaches we can take to prevent situations like this from happening again. But for now, I want us all to understand this technology rests within a broader system of healthcare and represented one of many layers of vulnerabilities in practice. So over the past few decades, not only have oximeters become cheap and convenient, finding applications in daily clinical workflows, they've even become affordable for home use. And as we expand the use of these devices, we risk furthering disparities in these populations. For some patients, they receive a false sense of reassurance that their pulse ox reflects a normal arterial oxygenation. Our ED received funding to distribute pulse oximeters to patients who were sent home to self-monitor, and some percentage of them likely should have returned to the ED for hypoxia that was never appreciated. What about the consequences outside of COVID? Patients with pneumonia or COPD may have had their risks underestimated, influencing decisions on whether to admit or divert home. Similarly, light-based technologies have been used in other devices, such as infrared thermometers, heart rate detectors, and non-invasive glucose monitors, all of which have been found to be error-prone in patients with darker skin tones. We can easily begin to imagine how the consequences of this technology's vulnerabilities pool over into other medical decision-making processes. In February of 2021, the FDA announced a safety communication citing the 2020 Michigan paper that I presented earlier today. They detailed a set of limitations for healthcare providers, noting that, quote, if a FDA-cleared pulse oximeter reads 90%, then the true oxygen saturation in the blood is generally between 86 and 94%. And they also emphasized that pulse oximetry is more valuable for assessing trends over time instead of absolute thresholds. When we are working up COVID, we are given absolute thresholds that make the difference between admitting patients for initiation of guideline-based therapy versus discharging home for self-monitoring with the risk of loss to follow-up. Here, the FDA is reminding us that the two standard deviations give us a range of 8 percentage points. This is significant. The announcement ends by stating that they're still evaluating the literature and that they may reassess the contents of this document. Of note, this communications page was last updated in November of 2022. So in the discussions that have followed, here are some of the ideas that have been raised in response to this problem. But as you'll notice, each of these is problematic in their own ways, but it leads us to a broader discussion around what to do with race in medicine. And so we'll revisit these ideas in a few moments. I'd like for us to visit this 2020 NEJM paper by Vyas et al. To explore the history and some up to very recent practices of using race in clinical algorithms. Now, had we not examined the role of race in this experience, we would not have recognized inequities with pulse oximetry use. However, there is a dangerous tradition of using race in medical decision-making in the United States, which historically has aimed to identify biological differences between racial groups. The authors state that, quote, despite mounting evidence that race is not a reliable proxy for genetic differences, the belief that it is has become embedded within medical practice, resulting in a number of unintended consequences. And it begs the question, when, if ever, is it appropriate to consider race in clinical decision-making? What can we do as clinicians to critically examine the role of race and ethnicity in medicine? So let's demonstrate this with an example from nephrology. We approximate kidney function using the estimated glomerular filtration rate derived from serum creatinine levels and demographic characteristics. One of these characteristics, the binary of black versus not black, results in a positive adjustment in the eGFR. Historically, the developers of this algorithm justified the creation of this race-based correction for Black patients on the basis of studies showing higher average serum creatinine concentrations among Black people than white people. They pointed to Black people being more muscular as the cause, which has since been disproven. In fact, the derivation studies were highly problematic. In one of them, though there were patients of all races, everyone was categorized as either Black or not Black, suggesting illogically that Black people were uniquely distinct from all other humans. Furthermore, the Black participants had excess diabetes, hypertensive kidney disease, and were classified as being in the lowest available income group, making this a poor study to generalize for the Black US population. So what are the consequences of this model? If Black patients are receiving overestimations of their GFR, this would delay a timely diagnosis of chronic kidney disease and delay their referral for specialist care and to transplantation. And this is already in the context of Black patients having higher rates of end-stage renal disease and mortality due to kidney failure compared to the overall population. So are these researchers justified in using a race-based correction? Historically, the origin of the use of race in medicine in the US is often linked to the Swedish botanist, zoologist, and physician Carl Linnaeus, who is acknowledged as the father of modern taxonomy. He used prior categorizations of humans into four major racial groups and began to assign them distinct skills, traits, and abilities, attributing desirable traits to white Europeans like innovation and civility, and least desirable traits to Black Africans like savagery and indolence. These attributions by a well-regarded physician established a scientific justification for biologic race and upheld assertions of racial superiority and inferiority. The rest of the medical community adopted these ideas and constructed an ideology of race-based biological disease risk. But since then, DNA studies have confirmed that all racial groups are genetically similar. But despite this knowledge that race is a social construct, it continues to be treated as a simple biologic variable. And no national or international consensus exists for defining race, which is usually determined by self-reporting or inferred based off of phenotypic appearance. The Office of Management and Budget, which determines how the federal government classifies race and ethnicity and informs the US Census, still uses many of the original Linnaean-based racial categories. This collection of data is vital in identifying racial inequities in the United States. However, advocates express it can also help perpetuate a racial caste system. In the 2003 JAMA Special Communication, authors Bennett and Kaplan illustrated the challenges in writing about race and ethnicity. To begin with, identity is not always fixed or easily categorized. A single race or ethnicity might work for some, while others may feel connected to multiple groups. Self-identifications can change over time as categories take on new social or political leanings, and the census categories we currently use are imprecise and mask significant heterogeneity. Secondly, it's important to distinguish between race and ethnicity as a risk factor and as a risk marker. For example, being in a certain group may be a risk marker for a condition if the prevalence of that condition is higher in that group. However, one's identification with that group does not itself constitute a risk factor for that disease. These racial associations with health outcomes should not imply causality and often reflect a range of social and structural factors we'll discuss shortly. And lastly, it's imperative that in using race and ethnicity, we do not further stigmatize populations or reinforce biases against these populations. But it's important to remember, without these categories, how would we effectively measure and appreciate racial disparities in health? How would we negotiate these needs against the critiques of applying race in the biomedical sciences? So in the example of nephrology, what is the role of race in driving higher levels of creatinine that the original paper hoped to correct for? We can see how structural racism and racial discrimination may have significantly contributed to the excess morbidity and poverty of Black patients in the derivation study, resulting in worse kidney function that presented as a higher baseline creatinine and risk for chronic kidney disease. But notably, European studies did not reproduce these same higher creatinine values in Black patients. But let's dig deeper and look at the broader constellation of factors of what that data represents. In healthcare, there is a strong body of literature arguing that for social determinants of health, intimately attributing a range of structures as causal factors in driving health outcomes. However, more recently, thanks to social, political, and academic activism, we are starting to internalize how deeper structural forces shape all the modern institutions which build, shape, and administer the environments around us. The institution of slavery began in 1619 and lasted formally until Juneteenth in 1865. After a decade of the Reconstruction era, Jim Crow laws cemented racial segregation until the Civil Rights Act in 1964 and Voting Rights Act in 1965. Since then, Black populations have been targeted by mass incarceration, racialized terrorism in the form of the KKK and white supremacy, and racialized capitalism as seen by exclusionary zoning, subprime mortgages, and more. And to give some reference, this is when America declared its independence, and here is when Highland Hospital opened its doors in 1927. And we are here in 2023. It's difficult to measure the effects of our histories on the health of our communities, but this is not just our history. It is very much our present. And as I've extended this box, it looks like it will continue to be our future unless we actively work to extricate these forces from our society. So we are taking complex social and political interactions that have been built off of the socially constructed premise of race and reducing the collective consequence of this oppressive ecosystem to just a categorical variable. This is a dangerous abstraction. By simply correcting for race in these prescriptive clinical guidelines, researchers risk interpreting racial disparities as immutable facts, rather than a series of injustices that each require intervention, and furthermore risk exacerbating these very inequities. Our data, including the racial differences within, reflect the lived experiences of those populations. And so when we correct for Black populations, we are actually correcting for the experience of systemic racism in America and risk reinforcing that disparity moving forward. So when should we include race? Going back to Vyas et al., they reflected on the 7 recommendations made by Bennett and Kaplan and adapted those guidelines into these 3 questions to evaluate race correction in clinical setting. First, is the need for race correction based on robust evidence and statistical analyses with consideration of internal and external validity and potential confounders and bias? Is there a plausible causal mechanism for the racial differences that justifies the race correction? And would implementing this race correction relieve or exacerbate health inequities? There are a multitude of rudimentary algorithms across medical disciplines that use to approximate racial patient risk, in which the use of certain racial categories leads to the systematic underappreciation of disease burden, shaping downstream flows of clinical attention and resource allocation. Here at Highland, we no longer use these adjustments, but these practices have not fully been universally reversed across the country. From cardiology to oncology, surgical and transplantation risks, are all of these adjustments justified? How are these guidelines impacting the way we determine appropriate diagnostic and therapeutic options for patients? I want to highlight the compounding effects that such race-aware practices can have outside of the bounds of our hospitals and clinics. During 2020, the CDC and the Department of Justice introduced guidelines acknowledging how having certain underlying medical conditions, including kidney disease, increased one's risk of severe illness or death from COVID-19, and they created a pathway towards a compassionate release from jail or prison. The courts reviewed applications using the race-aware GFR to determine kidney function, and as a result, numerous Black patients had their compassionate release applications influenced and determined on the basis of race. This meant that the Black patient, already potentially underdiagnosed and undertreated, would remain exposed to the unique risks of the prison system, including overcrowding, limited hygiene, and accelerated transmission of communicable diseases. To put this risk in context, the age-adjusted COVID mortality rate among prison populations was 3 times higher than that of the overall population. And then 2 more examples from litigation. During the massive class action lawsuits in the 70s that emerged after thousands of workers were exposed to asbestos, a race correction for Black and Asian patients was used in pulmonary function testing that led to an underestimation of lung damage and delays in diagnosis, treatment, and an attempt by employers to deny or reduce compensation for damages. More recently, in 2013, in the class action lawsuit against the NFL seeking compensation for cognitive impairment secondary to concussive head injuries, Black former NFL players were automatically assumed to have started with worse cognitive functioning than white former NFL players in a process called race norming. And by suffering less relative impairment, they would have been less likely to qualify for compensation. These examples show how race adjustments have been abused to deny Black people the appropriate attention, care, and resources they deserve. Looking outside the hospital, you will find proxies for race as well as race itself being leveraged across multiple industries. And I'd like to touch on some of these examples to illustrate the scale of our present and future challenges. Throughout these slides, I want to emphasize, if our underlying knowledge reflect racist social structures, then the reliance on racist outputs to inform predictive tools will only reinforce those very racist philosophies into policies and practices. So in health insurance, one company working with health insurers wanted to determine how to better allocate resources. These kinds of risk prediction tools are used on about 200 million patients across the country every year. By identifying high-risk patients, they could enroll them in programs with extra nursing support, PCP visits, and more. One of the largest of such companies used predictive modeling and took past healthcare costs into consideration to predict clinical risk and resource allocation. However, because the health insurer had historically spent more resources on white patients than Black patients, the tool predicted higher risk scores for those white patients, even when normalized by disease severity. As a result, it preferentially identified and enrolled far more white patients than Black patients into this care coordination program. The authors estimated that this miscalculation reduced the number of Black patients who should have been enrolled in high-risk care programs by more than half. If maintained, it would have worked to further deprive Black populations of appropriate care and worsened disparities by race. In criminal justice, these predictive tools have also been a particularly acute problem. Predicting recidivism, or the likelihood that a convicted criminal will reoffend, has been used to guide bail and prison sentencing for years. In 2016, a ProPublica investigation looked at Compass, one such recidivism prediction tool. The data used to inform this algorithm did not ask directly about race. However, it did ask about prior encounters with law enforcement, childhood upbringing, parental divorce, familial criminality, associations with friends who had been arrested, and even housing, education, job stability, and emotional attitudes were queried, all of which have been shaped by our histories with structural racism in the US. They found the following results. Black individuals who did not reoffend were actually twice as likely to be labeled as higher risk than white individuals. And white individuals who did reoffend were twice as likely to be labeled as lower risk as Black individuals. So even when we didn't ask for race, our data still reflected our history of racism. Now, generative AI tools have captured the public attention, and so I'd like to comment briefly on how they are built without us getting lost in the technical details. Large language models, the text-based tools such as OpenAI's ChatGPT-3 or Google's Palm or Lambda, are trained in string prediction tasks, which mean that they predict the likelihood of words in the surrounding context. Various technical tools are used to achieve this, from word embeddings to transformer models and different types of training and fine-tuning. You need a data set to train this model, which is done by extracting information about the relationship of words with one another. What changed though, in 2019, advances were made in transformer architectures that resulted in massive data sets being used to train even larger models, and an arms race began. So in 2020, GPT-3 was trained on 570 gigabytes of data that included 300 billion words. As a result, when prompted by text, the model is able to output impressive strings of coherent-looking text that was also incredibly racist, sexist, homophobic, occasionally calling for violence, and frequently producing falsehoods or hallucinating. Why is this? The truth is, these models don't actually have conceptual knowledge of the world or anything in it. When you prompt it for a recipe and it lists ingredients, the model has no understanding of what sugar, flour, or water are. It's merely trained on an immense corpus of text and is outputting a series of words that reflect the relationships of words in the text it was trained on. And given how large this model is, it's no wonder it can generate responses to an array of prompts. So it's important to acknowledge that these models have no capacity to understand what is true or false. This is why you cannot say that GPT-3 is lying, because lying implies a form of content. There is no intention with GPT-3. It is fundamentally a powerful statistical word predicting tool, which is why researchers have had to politely describe its outputs as hallucinations. As a result, these models have no understanding of what race is or what racism means. But by reflecting the world of text online that humans in mostly Western contexts have generated, the model forwards those word associations and repeats them in its outputs. You can prompt it to not be racist every time you use it, but you cannot rewire the statistical representations of words to disentangle manifestations of racism. So you are limited in the kinds of guardrails you can apply in building a safer model. And the same logic applies for image models, where instead of thinking about word relationships, it uses pixel relationships. As a result, it only makes sense for these models to produce visual outputs that reflect the racism seen on the internet. So if our underlying data on racial differences instead reflect the confounding effects of historic and present forms of structural racism, feeding them into future tools will only further cement racism into practice and policy. So I wanted to bring us back to the core challenge posed by Fawzi et al., that we predominantly use pulse oximeters with significant vulnerabilities across patients with different skin tones. Unlike the use of race correction in calculating GFR or the interpretation of pulmonary function testing, the race and ethnicity-based discrepancies of pulse oximetry exposes a fundamental flaw in the acquisition rather than the interpretation of data. This is because the differential accuracy of pulse oximetry is based on differences in melanin concentration, which does come down to a biologic difference. So what can we as individuals, as health systems, and as a country do to correct for this vulnerability? To frame these approaches, I'd like to start off with one of the arguments made by Dr. Ibrahim Kendi. An idea, action, or policy is either racist, that is contributing to a history that regards and treats different races as inherently unequal, or it is anti-racist because it is trying to dismantle that history. There is nothing in between. In this argument, he steps away from the idea of people being in a state of racism and asks us to evaluate the individual actions and tools we use. It is simply not enough to just not be racist. So when we choose to be anti-racist, we become actively conscious about race and racism and take actions to end racial inequities in our daily lives. Being anti-racist is believing that racism is everyone's problem, and we all have a role to play in stopping it. And to help operationalize these anti-racist ideals, I'd like to equip us with the idea of design justice. Design thinking is a human-centered approach to innovation that draws from the designer's toolkit to integrate the needs of people, the possibilities of technology, and the requirements for business success. However, whoever sets out to design enacts a monopoly over the creative thought process. It's these very dominant frameworks of largely white, cisgender male designs that have built our current institutions and have resulted in the exclusion of majority world populations on the basis of race, gender, ability, and class. As a result, we need to critically examine our tool sets and practices to deconstruct this dominant notion of design and ask questions such as whose perspectives informed these tools, who were they made for, and who was excluded from the narrative. Design justice is an approach to design that is led by marginalized communities and that aims explicitly to challenge rather than reproduce these structural inequalities. So this process starts with an ethical imperative to systematically advance the participation of marginalized communities in all stages of the technology design process. So as individual physicians, what can we do? How can we begin to rectify the challenges posed by pulse oximetry? One might ask, why don't we just rely on gold standard testing and get a blood gas? Arterial punctures are generally perceived to be safe procedures, but they're not without risks. A 2022 Dutch study reviewed nearly 500,000 procedures and found small but notable risk for complications that include embolisms, aneurysms, nerve damage, and more, presenting at a rate of 0.14%. If you have 100 patients who need ABGs, this complication rate may seem reassuring. But if we look at the scale of COVID, this begins to add up to significant harm. So we need to consider the increased exposure and morbidity that these complications would cause. Another path is applying some form of bedside adjustment for the measurement disparity. Like we discussed, the differential accuracy of pulse oximetry is dependent on the biological trait of melanin. And so instead of correcting for race as a proxy for skin tone, why can't we just adjust for skin tone itself? Inclusive tools for assessing skin tone are available, such as the Monk skin tone scale, but no proven methodology to correct the noise and variable bias of these devices currently exists. And these bedside adjustments definitely should not be left to interpretation by physicians on visual exam, which would create a new opportunity for bias. I think what we're left with is an important opportunity to re-examine how we treat high-risk subgroups in high-risk disease states. For multimorbid patient populations who straddle the boundaries for determining hypoxia or triggering interventions, we need to consider whether we should lower the threshold of receiving those interventions and leverage ABGs more frequently. Of course, this would expose them to the risks of those interventions, but if the alternate path is potentially underestimating the risk and denying them appropriate care, then we need to weigh these trade-offs. No matter how we change our practices, a justice-oriented approach presents an obligation for us as providers to invite and platform members of the community to these conversations and ask them for their input in helping us do justice to the people who will be most impacted by these decisions. Hospital systems so far continue to purchase and use oximeters despite their known vulnerabilities in non-white patients. The Federation of American Scientists, in a comment letter to the FDA, proposed system-wide structures to publish the real-world performance of all pulse oximeters used, stratified by device across high-risk subgroups. This data can fill the gaps in our understanding of current models and can be leveraged in contracting negotiations to discontinue underperforming models and procure devices that work more equitably across populations. They also considered new system-wide pulse oximetry thresholds to diagnose hypoxemia in darker skinned patients and account for the overestimation of these devices. Importantly, this would be adjusting for skin tone as opposed to the proxy categories of race and ethnicity. These interventions might improve care in the short term, but they don't solve the underlying problem of biased technologies. There's no guarantee that this process will uncover a sufficiently equitable device. We already have the technology necessary to detect melanin and adjust for skin tone, but regulatory reform will be critical in proliferating more equitable tools. And so it's important to recognize that health systems will have an outsized role in advocating for these reforms. So every new pulse oximeter that is approved by the FDA goes through a 510K pre-market notification process. This allows devices that have, quote, substantial equivalence or the same intended clinical use or technological features to a previously approved device to qualify for a fast track approval. Why is this? Current legislation requires the FDA to consider, quote, the least burdensome appropriate means to demonstrate a device's substantial equivalence. As a result, biases from earlier studies end up getting reproduced in subsequent products, and we don't end up seeing the full picture of the clinical data until years after a device has been approved. What did this look like for pulse oximeters? Well, in the 80s, the first oximeters had no requirements for gender and racial diversity. And after being adopted from a Japanese study, they were tested in the US almost exclusively on white male patients. Following the creation of the 510 clearance pathway, only 10 participants were required to demonstrate efficacy, and it had to include at least 2 darkly pigmented individuals for testing. This grossly misrepresents the US patient population. What does this look like for Highland Hospital? This is the pulse oximeter we distributed in our EDs on the left. And in their manufacturer's documentation, it reports only using those same 2 wavelengths of light and testing on only a dozen subjects with high variability and no stratification based on either race or skin tone. So regulators are in a position to ensure that pulse oximetry be tested across all skin types using objective measures of skin tone. Testing should occur across patients with respiratory and hematologic conditions and patients who have both acute and chronic presentations that can impact light absorption. They should also include bedside measurements of skin pigmentation to adjust the sensor or to assist in interpreting results in bedside. And these adjustments to oximetry should fit seamlessly into our clinical workflows so that we can directly compare melanin-adjusted pulse oximetry results across multiple patients and skin tones to allow for further analyses. More broadly, regulators have an opportunity to raise the threshold for FDA's evaluation of devices and algorithms. This means enforcing sex, racial, ethnic, and disability parity in government-funded research. African Americans represent 12% of the US population, but only 5% of clinical trial participants. Hispanics make up 16% of the population, but only 1% of clinical trial participants. And sex distribution in trials is predominantly male. The exclusion of women from clinical trials has led to a doubling of the rate of adverse drug reactions in women compared to men. Furthermore, medical diagnostic equipment is often not accessible to adults with disabilities. As it stands, diversity in testing is a voluntary guidance, and so our advocacy should center the voices of marginalized populations and make this diversity enforceable. Lastly, we should use disparities in health outcomes to drive research funding and auditing. Aspiring for equality is not enough. In order to seek justice, we need to prioritize identifying and rectifying the harms caused by our practices. In October of 2022, the White House Office of Science and Technology Policy released their blueprint for an AI Bill of Rights that laid out recommendations regarding protection from unsafe algorithmic systems, equitable designs of algorithms and protection from discrimination, transparency on how algorithms are being used and why decisions are being made, and the ability to opt out of algorithmic decision-making altogether, among other principles. However, these were non-binding recommendations, and so we need to ensure that marginalized populations not only have transparency, but meaningful mechanisms for accountability to seek justice. And so legislative efforts are still needed to enshrine these protections into law. You know, it would be great if we just had a better pulse oximeter that worked for all populations. But the surprising thing is, it already exists. So in the 60s and 70s, Hewlett-Packard worked with NASA engineers to create devices to monitor astronaut health. Precise measurements were key, and so they used fiber optics and 8 wavelengths of light compared to 2 today. They worked with 248 Black volunteers to better understand the impact of melanin on results, and it was also designed for the ear, given higher accuracy in patients who are septic or in acute disease, while also better accommodating gender and disability differences. And it had an option to sample a droplet of blood from the ear to calibrate the device by how much light was being absorbed by the skin. Unfortunately, when HP shifted to personal computing in the 80s, their healthcare device innovation slowed, and the lessons learned were never picked up by later developers who independently developed our modern pulse oximeter device. You can still go online and read their full report from their October 1976 journal, seen here. And here's a photo from the early 1970s of the HP pulse oximeter and it being used on someone's ear. Interestingly enough, this was actually used as a reference for some early pulse oximeters. And so I'd just like to summarize some of our findings today. Current pulse oximeters failed to account for melanin and resulted in the differential treatment of populations as measured by the proxy of race and ethnicity. As a result, vulnerabilities in our technologies magnified disparities in patient care during the COVID-19 pandemic, contributing to morbidity and mortality in already marginalized patient populations. A justice-oriented design approach can deconstruct these harmful dominant practices and elevate marginalized voices in creating more equitable tools and practices. We need to critically examine the inclusion of race in medicine using the 3 questions that we discussed earlier. And as long as our data on racial differences instead reflect the confounding effects of historic and present forms of structural racism, feeding them into future tools will only further cement racism into practice and policy. And so the revolutionary mindset of anti-racism is not simply about bringing harmful systems to an end, but it's about envisioning new ones. As we strive to make medicine a more anti-racist field, we need to ensure our practices do not perpetuate the very inequities we aim to repair. And with that, I'd like to extend my sincerest gratitude to the many advisors who helped give input into this presentation. And I'd like to open up our floor to questions. Thank you.
Thank you, Ajeet, for your presentation. It's been great and very eye-opening. So if anyone wants to ask a question or a comment, please unmute or write in the chat.
Hi guys, I'm happy to start. I'm just a visitor here, but Ajeet, that was really, really wonderful. Can you guys hear me?
Yeah.
Yeah, okay. As a interested future clinical trialist, I thought it was really interesting that you brought up the point that clinical trial patients don't really reflect the general population that we are aiming to treat. And especially in rare and ultra rare diseases, which is an area that I'm particularly interested in. That diversity exists within the very small population of patients, but we don't often choose them as our prototypic patients for clinical trials. As someone who's intimately involved in some right now, I can see the thought process behind our senior PIs when they're picking ideal patients. And when we talk about the ideal patient, they're often the ones who are going to what we refer to as be adherent and compliant to follow-up visits. Is there any way that we can incentivize policymakers to help, big industries and companies get financial support to assist in allowing us to, or encourage them to develop that diversity? How might we be able to encourage policymakers to incentivize big pharma and big industry to provide those resources for that diversity of patients who may have difficulties getting to appointments for follow-up, so they become a more ideal candidate for a clinical trial?
Yeah, Dr. Patel, I'm glad you brought that up. So definitely, it injects so many opportunities for bias when we try to identify who are the ideal patients, right? And that's informed in a context of not only our racialized histories, but also kind of wondering these patient populations themselves have histories, like very justified histories of distrust with the medical system. And so when we look at patient populations who we want to have a better job of representing, we need to find better ways for industry to not only incentivize, but really find ways of compensating or finding ways to help prove the value to these patient populations, that their involvement will help them, and that their involvement will not be an opportunity for exploitation, but rather an opportunity for learning and growth. And I think that there are a number of structural challenges in our private, our largely private but publicly funded research industries that need to seriously contend with their histories of dealing with these patient populations in order to overcome the challenges that they have likely contributed to themselves. So definitely an astute point. And I have another question in the comments from Dr. English. At least a couple of physicians have told me that pulse oximeters should be banned, not too practical. How do we advocate for improvement? Well, as buyers, our health systems stand as really important actors in deciding what kind of pulse oximetry devices are considered appropriate for our healthcare settings. And so if health systems as bulk buyers demand greater equity in the representation of their trials and demand better equitable performance by manufacturers, this will have to force manufacturers to reconsider how they are testing their devices and the technologies that they use. Again, these are not new technologies that have to be devised. We've invented these before. And so it's not a matter of trying to figure out this one quick trick. It's really a matter of shifting the industry to understand that equity needs to be a priority that we all need to invest in. And so that's why I say health systems stand as really key stakeholders in trying to advocate for pushing the window on this, pushing the needle on this.
We can also make better decisions soon, I think. The Federation of American Scientists has designed, and I don't know where they are on it, a large study to compare the different commercial probe types available on people with different skin colorations, and also to compare where they're placed. So not just the makers, but also whether it's used on the ear or the finger. Melanin concentrations differ by site of the body in an individual human, and blood flow does affect significantly the absorption as well. So hopefully in the next few years, assuming that a better technology doesn't make it to market, although which it certainly could, because you can do melanin measurement and correction as part of the algorithm within the pulse ox. And then there's other technologies that are looking at multiple different wavelengths, larger amounts of wavelengths, to actually solve this problem as well. But we will have better information as health systems to purchase. And I think we could advocate for that sooner rather than later.
Hi, everybody. I just had a quick question, if I'm audible. Hi, everybody. My name is Ishvir. I'm also a guest. I happen to currently work at Google, And so I think about a lot of the questions in the design team adjacent to your presentation. Thank you for it, Ajeet. I'm sort of thinking from a social cultural perspective. I mean, we all know that race is a social construct, and yet the demands of, I think, scientific research and testing sort of demand that we use, you know, the US Department of State's post-World War II categorization of the world, where Asia, representing a far greater percentage of the population, is a category that gets sort of like utilized in scientific research. And we know the sort of ills of that, even in like the Bidel case, for example, in terms of targeting and trying to ameliorate racial disparities in healthcare. I'm wondering if you've thought about the ways in which anti-racism means that we have to sort of deconstruct our idea of race in the first place when trying to address race as a problem, if you see what I'm saying. Like, if we presuppose these categories, how can we use these presupposed categories that are already themselves problematic in trying to address these sort of inequalities in the larger healthcare space? Thank you.
Yeah, I think that's a really interesting point, and that speaks to the fact that, you know, while these categorizations are problematic to begin with, only by utilizing these categorizations have we been able to measure the disparities between them in the United States or in these contexts. And so I think that does beg a broader question as to how communities want to themselves be identified and categorized. And by hopefully progressing to a more inclusive and pluralistic view of categorization for our populations in the US, can we then begin to imagine new ways of measuring disparities and being able to target ways to address the historic forms of disparities that have left so many of these communities behind or actively resulted in their exploitation and marginalization? And so there's not going to be any sort of single solution to addressing that issue, but I think, you know, a strong start would be just trying to find ways to elevate the voices of those who are marginalized, to try to ask them how they would want to be included and categorized, so that the inclusion is done on their terms as opposed to any sort of predatory inclusion.
Great. Thanks again so much, Ajeet. I'm sorry we run out of time. It's right 1 p.m. right now. Thanks everyone for joining again. That was a great talk. And yeah, if you have any other questions, I'm sure they know where to reach you, or if not, they can just let us know and we can send that talk as well. Thanks again, Ajeet. Great job.
Thank you everyone for coming.